Okay, good afternoon, everybody. My name is Brandon Bohr, and I'm an associate professor here at Purdue University in civil and construction engineering. And I had the honor of introducing our speaker today, Professor Vishal Verma from the University of Illinois Urbana-Champaign. And Professor Verma will be giving a webinar as part of our Purdue Institute for a Sustainable Future Impact Series webinars. And he will be talking to us today about novel tools to assess the health risk from outdoor and indoor air pollution. So Professor Verma is an associate professor at the University of Illinois, and his current research is focused on assessing the health risk of ambient air pollutants with a focus on particulate matter. In his 18 years of research career, Professor Verma has published more than 70 peer-reviewed articles in highly ranked journals, such as Environmental Science and Technology, and has presented his work in at least 60 seminars and conferences, included several invited talks. Professor Verma has earned numerous awards and recognitions for his research on air pollution, including a National Science Foundation Career Award in 2019, an honorable mention in the James J. Morgan Early Career Award from Environmental Science and Technology, a Center for Advanced Study Fellow, a top 0.5% scholar by ScholarGPS, and an Excellence in Review ES&T Air Award just recently in 2026. So thank you, Professor Verma, for agreeing to talk with us today, and we look forward to your presentation. After Professor Verma gives his presentation, we'll have time for questions from the audience. Thank you very much.
Okay, thank you. Thank you so much, Brandon, for such a nice introduction and inviting me for this wonderful series of the lectures seminar. So this I'm really honored to have and getting this opportunity to present my work here. So I'm on the slideshow mode. I think everybody can see my slides now. Okay, so the topic of my talk, as you can see, is the novel tools to assess the health risks from outdoor and indoor air pollution. Is it? Okay, yeah, the key was not working, so I think I'm just advancing the slides from my mouse. So this is an outline of my talk. I will first start with a brief background on the PM 2.5 toxicity. And since oxidative stress has been proposed as one of the major mechanisms responsible for the toxicological mechanism, I will talk about what is it this oxidative stress really is. Then I will talk about the various methods available, some of which have been developed in our own lab for measuring the oxidative potential, which is a close surrogate of the oxidative stress or the PM 2.5 toxicity. Then since oxidative potential has been linked with the chemical composition of the aerosol, I will talk about, from the studies conducted so far, what specific chemical components and their emission sources have been linked with the oxidative potential of the particles. And finally, I will also talk about the oxidative potential of the indoor PM, because that is an increasingly important source of the PM exposure. And how does our daily residential choices, this is based from our preliminary studies we conducted in the last couple of years, that they can affect the exposure to the toxic PM or the oxidative PM. I would like to first acknowledge the funding agencies who have supported my work so far, and my collaborators, and the wonderful group of students, my master's and PhD students. This is a very old picture, I think probably taken in 2022. I didn't get a chance to update it. Some of them have graduated from my lab, but some of them are still here. So really, this is their work, and you can see in my slides that the specific contribution from these students in different tasks. Okay, so as per the latest report from HEI, the State of Global Air Report, ambient air pollution is again the second risk largest factor responsible for 7.9 million deaths in 2023. So what does that mean? That number, To me, that number does not signify much until we actually put that into a context. So to put this into a context, 1 in 8 deaths worldwide is linked to the air pollution, which is a big number. If you think about it, and 62% of these deaths are attributed to ambient PM 2.5, while the rest 35% are attributed to household air pollution, while the rest 3% are from ozone. So these studies are great because they can actually show us the relevance of this problem, and the magnitude of this problem. But what these epidemiological studies do not tell us that is about the mechanism, like how these ambient PM 2.5 are basically creating these health effects. And that's a damn important question, because unless we understand these mechanisms, we won't be able to calculate the uncertainties in these deaths. Like, we won't be able to tell how accurate these estimates are. And this is some of the stuff which I'm going to touch in my talk today, that we really need to understand the mechanisms and the chemical components, which are responsible for these deaths. Okay, so to understand these mechanisms, we conduct toxicological studies. So I call that as a toxicological framework. And luckily, in the early 2000s, there have been several studies, the mechanistic studies, conducted by several toxicologists. This is primarily the work conducted in King's College of London and UCLA, like kind of seminal work, which basically took these PM 2.5 particles mostly from the ambient sources, and they exposed them to specific cells, and then they try to understand how these PM 2.5 particles, through a very mechanistic understanding, are doing to these cells. So they propose several mechanisms, there are a couple of processes, signaling processes. But the important thing, in all these, the one common thing, in all those proposed mechanism was the presence of this process, or the called the oxidative stress. So what is oxidative stress? In simple terms, oxidative stress is defined as the stress caused by the abundance of oxidants or the reactive oxygen species. Let's try to understand it, more clearly, because this is where we are gonna actually spend in the next couple of minutes on that, and in fact the next hour, So I think let's try to understand why this ROS generation, or the oxidative stress, is important. So the first question I would like to ask is ROS really bad? Or always bad, or is it foreign phenomena to the body, or is it what is it ROS? Where do they come from? So to understand that, we have to really dig into some of our basic fundamentals. You all know the equation for the aerobic respiration. For example, whenever we eat something, glucose, it is oxidized by our body to generate carbon dioxide, water, and energy in the form of ATP molecules are released. That's a very simple equation, right? Everybody knows that. But if you try to remember your high school chemistry, and go a little bit further into that, it's a little bit more complicated process than this simple equation. That glucose first undergoes glycolysis to form pyruvate, which is then degraded to acetyl coenzyme A and carbon dioxide. Now, this acetyl coenzyme A then goes to the mitochondria, which is called the powerhouse of the cell. And then it undergoes this Krebs cycle, where it generates these two antioxidants, NADH and FADH2. The main purpose of these antioxidants, or the reductants, is to donate electrons, which are then transported through this complicated electron transport chain, at each step of which a fixed number of ATP molecules are released. So the last molecule which accepts this electron is oxygen, which in presence of the hydrogen ions, protons, which are abundant in the mitochondria, form water molecule. So you can see that how this equation is basically undergoing this process to release water, carbon dioxide, and ATP. So this is a normal process. But as you know, that nothing in the nature is absolutely perfect. So is true with this process. What happens that in 0.1% of the cases, that these electrons are prematurely accepted by oxygen, and if in the absence of hydrogen ions, like that it kind of missed that reaction, then oxygen directly takes up this electron to form superoxide radical, which basically has an unpaired electron. And because of the presence of that unpaired electron, it becomes super reactive. And that's why we call it as reactive oxygen species. So in any oxidative process, this is the first step of this ROS cascade, that the generation of superoxide radical. Once the superoxide radical is generated, then it initiates a series of chain reaction by which it starts forming other oxidants. So I will talk about that in a minute. So what we learned here in this slide is that ROS generation in the body is not a foreign phenomena. It is not like that, only if you are exposed to the PM or something like, that you generate this excessive ROS. ROS is a very natural phenomenon. In fact, as we eat our food, ROS generation is going to happen. In fact, ROS actually is a good signaling molecule. Like, it tells the white blood cells to, about the foreign invader, like, if in terms of the bacteria or pathogen, like, any pathogens. So if ROS are getting generated in the body and they are so reactive, they can kill the cells also. So there should be a auto defense mechanism also in the cell, because nature has tried to, like, design us that way. So in fact, there is, like, every cell has a pool of antioxidants, which can be of different type, but they're all characterized by their capability to donate electrons. So whenever a free radical or an ROS is generated, which has an unpaired electron, these antioxidants donate their electron and quench it. So it kind of completes this outer shell of this free radical. So that, it's ROS is gone now, which is, so I mean, no problem, right? And in a cell, there is a complex play between the ROS cascade in the antioxidants. If you see, like, oxygen, by accepting electron, forms superoxide radical, and then by accepting one more electron, it can form this hydrogen peroxide, which in presence of the metallic ions such as iron, copper, like, can generate hydroxyl radical. And then hydroxyl radical is way more reactive than the superoxide radical. So, this is called the Fenton reaction. And then when we have these antioxidants, such as, like, glutathione, which is a major class of antioxidant present in our cells, then this radical is quenched, like, to form water molecule. Or, if glutathione gets a chance to react with hydrogen peroxide directly, it can quench that to water also. So you see that antioxidants and ROS are playing in tandem, like, in fact, in a healthy individual, there is a perfect balance between the concentration of ROS and the antioxidants. So there is no problem in a healthy individual. The problem arises when there is a disbalance. So meaning the concentration of the ROS could exceed way beyond the capability of the antioxidants to support them, or the concentration of the antioxidants level, the pool of antioxidant, can deplete to a level that even the normal ROS generation cannot be supported. Both of these conditions are called oxidative stress, because what you have now is the abundance of oxidants, which can be counterbalanced by the antioxidants. Okay, so I think you understood that, what we having, like, what is this oxidative stress? Now, one question is left: what is the role of particles, like, ambient air pollution, in creating this imbalance between the ROS and the antioxidants? So let's try to understand this. So there are three modes prevalent, like, by which this ambient PM or other PM can actually induce this oxidative stress. One mode is that these particles can carry the ROS species on their surface. Like, for example, hydrogen peroxide could be in the particulate form. Hydroxyl radical could also be, but, it has a very short life on a scale of nanoseconds. So that is not a worry. But, superoxide radical, other organic peroxides can be present on the particle itself. And when we inhale them, they directly go to our lungs. So that is one mechanism by which they can enhance the concentration of ROS in the body. The other mechanism could be that not the ROS, like particles are not carrying the ROS, but they are carrying certain chemical components such as copper, iron, quinones. We call those substances as redox active substance. What these species can do, they can transfer when they are inhaled in our body, they can transfer the electrons from these electron donors such as glutathione, ascorbic acid, NADPH to oxygen. And once this oxygen accept one electron, they can generate superoxide radical. So they are basically doing this catalytic reaction, transferring the electrons from cellular electron donors to oxygen and thus generating the ROS. So they are generating ROS in situ in the body, which can generate more ROS, And then the third mechanism is cellular mechanism. Now that mechanism is more complicated. How does it work? We don't know exact, fundamental of that, but we know that certain biological cells, such as macrophages, epithelial cells, when they identify any foreign substance, such as particles, pathogens, bacteria, they try to engulf them. And in a process to engulf them, they generate reactive oxygen species which act as a signaling molecule to call the white blood cells to arrive at the sites. So these ROS serve dual purpose: oxidize some of these components as well as invite these white blood cells to come to the sites to engulf them. So certain particles can act as those foreign material, so these are the three prevalent modes of the oxidative stress, caused by the ambient PM. So now we understand how these process induce oxidative stress. Accordingly, scientists have developed different methods, chemical methods or cellular methods to simulate these mechanisms in vitro. So one common assay, which has been used very widely in the field of oxidative potential, is called the DTT assay, which is a chemical assay in which we take this compound called DTT, which basically represents the thiol group of antioxidants or the cellular reductants, and we incubate these particles with DTT. So what happens? That certain chemicals present in the particles can transfer these electrons from DTT to oxygen, and thus generating superoxide radical. And in the process, the DTT gets oxidized to ignore this oxidized DTT. So what we measure here is the rate of consumption of DTT, which is directly proportional to the rate of formation of superoxide radical. So you can see that what we are doing here in this process, we are basically simulating the process present in the mitochondria, the NADH driven ROS generation. In another mechanism, to simulate the cellular generation of ROS, we can take these cells such as alveolar macrophages or epithelial cells, and in them we inject a dye. I mean, one common dye is DCFH, or there are other dyes also available such as Amplex Red. Basically these dyes are non-fluorescent at the beginning. And what happens when we incubate these cells with the particles? These particles generate reactive oxygen species, and then these ROS reactive oxygen species oxidize this dye to another compound which is fluorescent. So the cell becomes fluorescent, and then we can count the fluorescence intensity, which is proportional to the rate of formation of the ROS. So these are these were the prevalent mechanisms, but they were lacking one dominant mechanism of the ROS generation, the Fenton reaction. As you might remember, that hydrogen peroxide in presence of the metallic ions such as iron, copper can generate hydroxyl radical. So that mechanism was actually lacking in the chemical assay. So to complete this whole process, we developed a new method for measuring the ROS generation, which is based on the hydroxyl radical generation. So what we did here is that we, in the reaction vial where we are measuring the superoxide radical generation, we added another quenching compound called the TPT. Now this TPT reacts with hydroxyl radical and then becomes fluorescent. So by using a spectrophlorometer, we can measure and it's very specific. It reacts only with the hydroxyl radical. So so by measuring the fluorescence intensity, increasing fluorescence intensity, we can measure the rate of formation of the hydroxyl radical as well. So by completing this process, we can basically measure both the superoxide radical as well as the hydroxide radical generation. Now, I would also like to explain, these two different expressions for representing the OP of the ambient PM. One is called the intrinsic OP because, has been a lot of confusion in the field, how we should represent the OP, the oxidative potential. So one expression is intrinsic OP, which is given by the OPM or mass normalized OP. So let's say we get the raw results of the oxidative potential, which is in the rate of consumption of DTT or the rate of formation of superoxide radical, hydroxyl radical, whatever ROC you're measuring. If you normalize that with the mass of the particle collected, it is called the mass normalized or the intrinsic OP. But if you normalize it by the volume of air collected, it is called the extrinsic OP or the volume normalized OP. How are they related? If you multiply the mass normalized OP with the mass concentration of the PM, it gives you the volume normalized OP, right? So why, in what context they are important? Intrinsic OP is a intrinsic property of the particle, which is a sole function of the chemical composition, right? So it is very useful while we're comparing the intrinsic toxicity of different emission sources, like which toxic, whether vehicular source is more toxic than the biomass burning, or like that. And extrinsic OP is more important because remember that it is given by it is a function of both chemical composition and the amount of PM in the air. So it is important when we are assessing the overall exposure of PM to the humans, like what is the total toxicity imparted by a given source to the humans. So we will be using these expressions in the next couple of slides. That's why I wanted to explain these things at hand first. Now, I would like to show some of the results obtained from our lab, because as I indicated that, OP, like this oxidative stress, has been proposed as the major hypothesis, but there was not a concrete evidence that it is playing a role. So recently we conducted a study on a very large number of samples collected from the Midwest US, about 300 number of samples collected from Bonneville, which is a rural site near Champaign, Chicago, St. Louis, Champaign, our hometown, and Indianapolis. And on these PM samples, we exposed them to the A549 cells, which is the human lung epithelial cell line, and we measured two endpoints, cell death and the cellular ROS. And you can see at all these sites, the cellular ROS generation showed a very consistent correlation with the cell death. So this is cell viability, so it is decreasing. So as the ROS is increasing, the cell viability is decreasing, right? At all these studies showed a very consistent correlation. But as we have seen this kind of correlation in other epidemiological studies also. But what's the point? How is the mechanism? How do we prove that it is really killing the cells? Well, to further confirm that, what we did for the first time, we incubated these cells first with the antioxidants, which are present in the cells, before exposing them to the PM. Okay, so what we did is we pre-treated these cells with glutathione. So we artificially increased the concentration of glutathione in these cells, and then we exposed them to the PM. And you can see that as we pre-treated them with glutathione and then exposed them to the PM, the cellular ROS generation reduced consistently in all these samples. And similarly, not only the ROS generation reduced, but the cell viability enhanced. So it is able to live longer. So this is kind of the proof. We showed to concretely establish oxidative stress as the major mechanism responsible for the toxicity of the PM. So we have developed several high throughput automated instruments in our lab. For example, this instrument measures the DTT activity of the particles in a completely automated manner, because the measurement of this oxidative potential is not straightforward. It is very laborsome process. So, and then my student Joseph Putusari is actually has also developed a real-time instrument for measuring the DTT activity. And then my other student Horan, you has also actually measured, has also developed this totally automated instrument which can measure the ROS activity of the particles by almost all endpoints available, such as glutathione consumption, ascorbic acid consumption, hydroxyl radical generation, DDT consumption. But what was lacking is the cellular generation of ROS. So my student Sudhir Salana has actually developed this instrument which can measure the ROS activity in epithelial cell lines in a completely automated manner. So basically, these instruments are critical that, now we can generate huge data set, like, on the measurement of the ROS activity, which can then be fed to the epidemiological studies. And with these instruments, what is the overall philosophy of our research lab? Like, we collect particles from various sources, such as primary particles, secondary particles, biomass burning particles. And recently, we have also started collecting samples from indoor sources. And we measure, the chemical composition of these particles in a very comprehensive way, and also measure the reactive oxygen species. And then first we try to use, like, this regression approach or machine learning models, like now recently that, because we are able to generate large data set, how these different chemical components are statistically related with the ROS generation. And after we establish those correlation, what we do is that we try to, like, break these particles into different chemical composition, like fractionate these particles into different chemical components, such as inorganics, organics, metals. And then we see how does the removal of a specific component affect the residual ROS activity of the particle. And based on this correlation and the mechanistic links, we try to predict the health effects. And then we link them back to their emission sources. Which emission sources really is most critical to the health effects. So this is like the approach my students have taken, like, collecting samples. I think this is a site from Indianapolis. We fractionate them into using the solid phase extraction column, measure their ROS activity, and then, this shows that how the cells look in a negative control, very happy cells, meaning a large concentration of the cells. But when you put the PM, how do they look like? They're mostly gone. And this is also, I think I would like to briefly explain about the fractionation scheme developed in our lab to segregate these particles into different subfractions, mostly the organic and metals. Like, so when we take this PM 2.5, we extract them into water, and when we pass them through a column called the C18 column, the hydrophobic organic compounds are retained on this column here. So the effluent of this contains, so the effluent here contains hydrophilic fraction, which is aliphatic substances, and the aromatic substances, or the hydrophobic organic compounds, are retained on the column, which we can elute using methanol or some other solvent. So in the eluate, we can recover this organic hydrophobic fraction. Now this hydrophilic fraction contains organic compounds plus metals. When we pass them next to the Keyless column, now this Keyless column can retain the metals. So like it can bind with the transition metals. So the effluent will be containing just the hydrophilic organic compound, and the metals which are retained on this Keyless column, we can elute them using a strong acid such as HCl or H2SO4. So this elute will contain metallic hydrophilic fraction. So you can see that how did we actually separate these different chemical components, meaning organic hydrophobic fraction, which is mostly the aromatic substance, which are also called the humic-like substances, which because they represent the humus present in natural water and soil, like, and aliphatic substances, aliphatic organic compound, and metallic fraction. Okay, so this is critical because this has helped us substantially to understand the roles of these. Like, for example, when we remove this specific component and when we measure the ROS activity, what is the effect of removal of that component? Like, it has helped us substantially. So in the last 2 decades, like, we have conducted a lot of studies using both statistical approach as well as this mechanistic investigation. So here I'm showing only the studies conducted in our group, but there have been other studies which have been very much in agreement with these studies. It's a busy slide, but what I want to give you the gist of that, by using this using high throughput instrument and this mechanical separation of the particles, what we have found is that two major classes of the PM compounds. Like, what are those compounds? Hulis, or the humic-like substances, and transition metals are the most important culprits for the ROS generation. As you can see, like, for example, this M by Z, which is oxidized organic aerosol, is highly correlated with the DTT activity. Same here, this correlation. And if you look at here, like, a lot of activity is really correlated, is really associated with this MOOA, which is like, if you know the terminologies of the this HRAMS, basically this is the Hulis compound, like, the humic-like substances. And then we further segregated this Hulis into different fractions, and we found that most of the activity was related with the the fraction which came off in methanol, which is having a lot of Hulis compounds, like, um, same thing, like, in most of these studies we found that metals is another class. As you can see here, that metals is really correlated with the ROS. Like, same here, metallic hydrophilic fraction was actually associated with a lot of ROS activity. So this is a gist, like, from these plots, like, from these studies, that humic-like substances and transition metals are the most important culprits for the ROS generation. Okay, so we now know that, okay, these chemical components are important. So can we just estimate the ROS activity if we are measuring these chemical components? It turns out that it's not that simple, because we found out in our later studies that there are quite strong interaction and mostly synergistic interaction between these different components. And how did we reveal those? So, like, what we did is that we, for example, we measure we expose these cells to the Helios type compound, and we measure the LC50. You know what is LC50, right? LC50 is a lethal concentration which is able to kill 50% of the cells, right? Now, it's a reverse parameter. So you can't add LC50 or two different components, right? So what we do, so we cannot understand if there are synergistic or antagonistic interaction if we just simply add or subtract these, right? So we measured the LC50 of Hulis, and then in that same setup, we added another component such as iron and copper or manganese at a non-toxic level, which we know that it is definitely not toxic at that level, like, and we see how does this adding a non-toxic level of that other component reduces the LC50, like, or affects the LC50. If it is reducing the LC50, that means there is a synergistic interaction. If it is increasing the LC50, that means it's an antagonistic interaction. And based on that, we found that when we mixed Hulis with any of these metals, we found a synergistic interaction, meaning these metals actually are able to combine, when combined with these organic compounds, are able to enhance the toxicity of these particle components. So what is the mechanism? There are several mechanisms, I think, which have been proposed in the literature, as well as, we also found new mechanism. One mechanism is that, the antioxidant present in the cells, or the reductants, when they donate this electron to oxygen to generate superoxide radical, and quinones, organic compounds like Hulis, are very active in these reaction. But the other ROS cascade, which is like, the generation of the hydroxide radical, it is mostly driven by the metals. So when we have actually this combination of quinones, or the organic compounds, and metals, we see a synergistic interaction, because we see the generation of both superoxide radical as well as the hydroxide radical. So that is one mechanism. Another mechanism is that, certain metals such as copper, iron, they are sensitive to, like, their solubility is sensitive to the pH, which is modulated by sulfate. So sulfate is able to, reduce the pH, and that's how it is able to actually make some of these metals more water soluble. And because they become more water soluble, they become more bioavailable to these species. And the other mechanism which we recently found out, that certain metals could complex with these organic compounds, and thus the solubility of these metals actually is way higher than if they are individual, like, the solubility of these complexes. So thus they become more bioavailable. So what, irrespective of the mechanism, we found that there are synergistic interaction between these chemical components. So estimating the ROS activities through simple addition, like, regression model, is not that simple. That's why we are actually moving on towards the machine learning approaches, which can actually model the non-linear interaction as well. So having these tools at hand and developing these skills, we launched our intensive sampling campaign worldwide, in which we collected samples from various sites in the world, such as India, Europe, America, South America, North America and South America. And for the first time, we measured like a very comprehensive suite of toxicity endpoints. You call it like DDT assay, hydroxylytic agenation, glutathione assay, cellular ROS activity, cellular death, all these endpoints. And we measured them, of course, the PM mass. So the objective was we wanted to understand, do PM mass and toxicity track each other? So this is the most comprehensive data set I think we obtained from all these sites, which shows very interestingly that all these endpoints such as DTT, ROS, GSH, hydroxylytical, cell death are actually correlated with the PM mass. So in a way, it justifies our standards, which are based on the mass of the particles, that as the mass increases, the toxicity increases. But wait, here's the catch. Look at the slope of these different these plots. For example, if you look at the slope of the ROS between ROS versus PM 2.5 mass, at Chile it is 5.37, while in UK it is 24.67. Similarly, if you look at the slope of the cell death, it is 1.28 in Chile and 3.89 at UK. I mean, you can see more variation if you look at, if you include the India data, so what does it show basically? That yes, toxicity increases as the PM mass increases, but not in the same proportion. And when we combine all this data together, what you find out is a supralinear relationship, meaning at lower concentration, it follows a linear trend, but at higher concentration, it plateaus. And if you look at the finding from the neuroepidemiological studies, they are also indicating the same thing, that the relationship between the mortality and the PM 2.5 health effect is not linear, it is supralinear. But they are not able to provide the mechanistic explanation for that. And I think these results provide a mechanistic explanation that why we are seeing this plateauing curve. So it basically shows the importance of measuring the intrinsic oxidative potential of the particle, which is a function of the chemical composition. Okay, so one obvious derivation of that, if you are measuring if the ROS activity or the toxicity do not track each other, then if you look at the emission sources, are the same contributing to the PM 2.5 mass versus OP? So we did we based on the data we collected from several campaigns, we basically estimated the contribution of these different emission sources to oxidative potential versus PM 2.5 mass. So this is the data which shows from this collected from the Southeast US, and you can see that there is significant disparity. For example, biomass burning contributes most significantly to the oxidative potential of the particle, 35%, but its contribution to the mass is very little, 14%. The other contributor which actually contributes a lot to the mass, secondary sources, do not contribute that much. I mean they contribute, but not in the same proportion as to the mass. So this is the data from the Southeast US, and this is the data from several sites in the Midwest US. This is Bondville site, rural Midwest, roadside, urban Midwest, which is St. Louis, Indianapolis and Chicago. And what we found, that the dominant sources contributing to the PM 2.5 mass are secondary sulfate, nitrate, biomass burning, and coal combustion, while the sources which are contributing to the cellular OP are secondary organic aerosol and agricultural activity. Surprisingly, in Midwest US, you look at this plot, 62% of the OP is contributed by the agricultural activity here, which contributes only 12% to the PM mass. And the dust, in certain sites such as roadside site. So you see substantial disparity in the sources contributing to the oxidative potential, toxicity, and the mass. Same thing we found in our campaign conducted in India as well. There are, I will just focus on this plot. The contribution of biomass burning and secondary sulfate to the oxidative potential is 37 and 34%. based on the previous studies conducted, we didn't conduct, like, we didn't apportion the contribution to the PM mass, but we found that the contribution of these two sources is pretty low, like, less than 30%. So, okay, so these are the results collected from the ambient PM. Now I'm gonna switch gear to the indoor air quality, because, as you remember that in the introduction I showed that indoor air pollution is also responsible for a majority of the deaths. As we know that people spend 85% of their time indoor based on this human activity survey, and more than 4% of the global deaths are actually attributed to the indoor air quality, from the hazardous air pollutants generated in the coming from mostly from the developing countries. So although the toxicity of, or the OP of the ambient particles has been well characterized, as you can see, like, the number of publication from 2019 to 2023, like, there is a ample data on the oxidative potential and toxicity of the ambient particle. The indoor PM has been rarely characterized for the oxidative potential of the toxicity. There have been only very few studies which are also scattered, like, somebody's measuring in China in homes, like, or somebody's measuring in Taiwan, or some, like, in US, like, very, very disparate, like, very scattered studies. So to understand systematically, like, how do these the particles coming from these indoor sources contribute to the OP, we characterize that using a like a well controlled environmental chamber, which we developed in our lab. We basically, like, it's a stainless steel chamber where we, like, is well characterized for its air change rate. And then we, like, for example, we put our source here, and then we pass this HEPA filtered air, and then we collect the exhaust and characterize that using SMPS, OPS, and collected the filters onto Teflon and quartz filters. And for site segregated analysis, we put cascade impactors, UTAS cascade impactors, to basically understand the size resolved chemical composition as well as the toxicity. So here are some of the data which we got. So we tested some of, like, we tested these sources, which are candle in presence of the wind draft, incense, cigarette, like two different type of incense, humidifier running on tap water or DI water, toaster, and this air fryer. And what we found is that the candle in presence of the wind draft showed the highest DTT activity and the hydroxyl radical generation. Cigarette showed the highest glutathione consumption. In general, combustion sources showed higher activity, like intrinsic oxidative potential, than the other sources, such as, like, the heating based sources such as air fryer or the evaporative based mechanism. So here, air fryer, toaster, humidifier, as well as the ambient PM. And interestingly, many of these sources actually showed higher intrinsic OP than the ambient PM. You can see this is the ambient PM, and then several sources actually showed higher oxidative potential than the ambient PM, which is significantly important finding. Like, why is this significant? Like, when we actually calculate the 24-hour inhalable dose of the OP from these sources based on the typical duration of these, typical usage duration of these sources, such as humidifier, like, we generally turn it on during the night for 8 hours in a closed room. Like, the concentration of the OP could exceed, like, could actually become significantly higher. Similarly, like, when we run these different sources for a typical duration, such as toaster, like, somebody toasting 6 or 7 bread. Similarly for the air purifier, air fryer. So we used a hypothetical exposure scenario that, a room volume of 50 meter cube, inhalation rate of 8 lpm, and the room ACR is 0.4, which can be debated. Like, what we found is very interesting, that the highest OP exposure, meaning the total dose of OP contributed by these different sources, is comparable between cigarette, humidifier, candle, like, because the duration, the usage duration of humidifier is much larger in comparison to the cigarette, right? And for almost all of the sources, the inhalable OP exposure, the OP dose, is either equal or exceeds way beyond the ambient PM exposure. Like, sometimes it can actually like go to 21 times of the ambient PM. So ambient PM is here, and you can see that for most of the sources, it is actually exceeding way beyond the ambient PM. And we also wanted to understand, okay, where these particles mostly contributed, like mostly concentrated to, like what's size range. So we collected some size result data also, which we are currently in processing this data. So here we showed the size result particle emission rate. So it's just a mass, mass of the particles emitted in different size ranges. And this is the size resolved intrinsic OP. So mass normalized OP in different sizes. So as you can see that the most of the particles are getting emitted in the smaller size ranges, but the OP, the intrinsic OP is kind of flat, but it's still higher in the smaller size range, significantly higher in the smaller size range. If we multiply these two terms, meaning the size resolved particle emission rate with the mass normalized OP, what we will get is the OP emission rate. So this is the emission rate of oxidative potential from these different sources. And you can see that most of the OP is actually coming from the smaller size range, less than 250 nanometer, which has a very high deposition efficiency. And because of this synergistic effect that most of the mass is in the smaller size range, which has interestingly, the highest intrinsic OP, the OP emission rate, it get more pronounced in the smaller size range. Okay, so this was based on the very controlled studies. And to further understand that, how does the real, how does the OP in the real home looks like? We also sampled several homes in Urbana, Champaign. So we basically recruited 21 participants. This is a small sample size. And then we visited them for for a week, for 6 months apart, study. And we measured the oxidative potential of the particles collected, and also ask the participant different questionnaires, like do you use candles, what is which type of stove you use. So here I would like to show some interesting relationship. I think I will just skip this slide in the interest of time, but here's the interesting part, that what we found, that the DTT activity and all the other parameters such as particle mass concentration, water soluble organic carbon, copper, manganese, were associated with the floor area, meaning larger is the floor area, smaller is the concentration, lower is the concentration of these species. Okay, and what is the effect of the cooking stove type? What we found is a univariate correlation. So the sample size is small, but still significant. So what we found that the homes which are having gas stoves were having significantly higher DTT activity as well as the particle mass concentration, in combination with the other species, such as, we know that water soluble organic carbon, iron, manganese are all redox active toxic species. As I showed you, these are the species which are really correlated with the RO estimation. And finally, the scented candle use, it showed higher activity, the homes which are having which responded that yes, they used scented candles, they were shown to have the higher OP as well as the PM mass concentration in their homes, along with the concentration of this water soluble organic carbon, copper, and manganese. So basically, this is a suggest that indoor origin particles plays a very important role in determining the OP exposure to your lungs. So with that, I will conclude, that the chemical components and the emission sources which are contributing to the PM mass are not necessarily same as those contributing to the health effects. So using this globally generalized linear concentration response, which are basically using the health effect as a sole function of the PM mass, could lead to unrealistic estimation of the mortality and morbidity. So we should try to include some sort of the intrinsic toxicity of the particle, to which oxidative potential could actually serve as a proxy. But I recommend more worldwide studies should be conducted to better understand its spatiotemporal distribution and its health relevance. And finally, the indoor PM, the the sources which are contributing to the indoor PM can release some reactive species which can exceed the exposure to the oxidative potential way beyond the ambient PM exposure. And your choices, like, such as flow, like, the source use characteristics such as scented candles, gas stove, I mean, can make a significant difference in your exposure to the oxidative or the toxic PM. I will stop here. Thank you.
Thank you, Professor Varma, for your very nice presentation. It's quite fascinating, especially the results that you shared regarding the indoor oxidative potential for different indoor sources of particulate matter. I think that's really very, very interesting. So for the
Thank you, Brandon. I should just
participants,
disclaim that it's a very small sample size, so we should just take it like in. I think that's what we concluded in that paper also, that we need to conduct more studies with the larger data sets. Yeah.
oh yeah, but I think really great work with the chamber measurements, the field measurements in homes. Quite fascinating. So for the participants, if you have any questions, can you please type it into the Q&A box from Zoom? So I think just to start, Professor Varma, I think in the United States, we focus more on outdoor particulate matter monitoring, and we have regulations and exposure guidelines for ambient PM. But as you have shown, indoor PM can drive much higher oxidative potential emissions and exposures, especially aggregated over a full day. So what are your thoughts about kind of the future of ambient and indoor PM monitoring and exposure regulations and so forth? Should we be thinking more about indoor sources of particles given their potential toxicity? And what could we do potentially to reduce some of these adverse health outcomes that you have observed?
That's a great question, Brandon. And so, I went to Indoor Air Conference in 2024, and I think as you saw in my slide also, there has been ample data showing in the ambient PM for the toxicity or the oxidative potential of the particles, and like the chemical components which are driving the response of the oxidative potential have been well characterized, but no such data actually exists for the indoor PM. So I think there have been ample data on the concentration of the particles from these sources, like for example cooking, and there is some chemical composition data also, size resolved data also, but not that much. But the toxicity data is almost lacking, so I think the indoor community is kind of is at a point where they're kind of stuck in, that okay, they're all emitting, I mean these sources are emitting all sorts of particles, but are they really health relevant, and how can we actually assess their health impacts? So I think if you ask me, my strategy will be to really conduct the studies on understanding the toxicology of these particles and relating these toxicity with the specific source specific chemical components. So that should be the way forward, so that we can really segregate, uh like which source is really important, and based on that, we can, the typical usage duration, we can really estimate these exposure. So I think right now we are lacking that data, and this is what we should be moving towards,
Thank you. And I think the size resolved OP emission rate metric that you came up with will be very important moving forward for characterizing the potential toxicity of different sources. I think that was quite novel. I've not seen that before. We have a few questions from the audience. The first one is, thank you for this very interesting presentation. You focus on particle mass concentrations regarding the toxicity of ambient and indoor particles. But what about the particle number? Have you or do you plan to study more the ultrafine fraction of particle toxicity, I suppose, with respect to particle number-based concentrations?
Yeah, that's another good question. So, so this is a very like how how to like how do these physical property and the chemical composition play a role? I would like to explain it this way, that the physical component, like the physical property of the particles, might not do anything, might not be related with the toxicity itself, because toxicity is the is really related with the chemical composition of the particle. But that doesn't mean the physical property is not important. Physical, like the size of the particle, actually determine what chemical components will be present. So, like for example, the certain chemical components might be concentrated in the smaller size ranges. So in a way, in an indirect way, particle size could be related with the toxicity. And so that is one mechanism. Another mechanism is that the particle size also determines how deep it can go to the lungs. As I showed, in the size segregated particle collection from the indoor sources, most of the OP was concentrated in the smaller size ranges. Now, where does it end up? It basically depends upon the size, right? So, and then what it does it do there, then it depends upon the chemical composition. So the both are important, but toxicity, like the way we are measuring the toxicity, we are making it a function of the chemical composition. We are not studying the dosimetry, how does it deposit in different parts of the lung, so where it can become important. And yeah, I hope that answered his question.
Thank you. Next question is, you showed the correlation between oxidative potential and particle size for indoor pollution sources. Have you considered doing that for outdoor particles instead of just total PM 2.5 mass? Do you think that could help explain the variability in slopes between sample locations across the different
Yeah.
measurement sites around the world?
Yeah. So I think that's another related question and important. So yes, it has been done for the ambient PM, and interestingly, all of that data showed higher OP in the smaller size ranges. I mean, we didn't do it in our studies because it has already been done by various groups such as UCLA, I think, and people in Netherlands. They have collected the size segregated particle, uh, you know, and then measured their OP, and they all showed similar results to the indoor OP. In fact, they were more pronounced in the ambient PM. I think what we showed is a kind of a flat, but with only slightly higher up in the smaller size ranges. But what they showed is a very sharp gradient, that the smaller particles were having much higher oxidative potential than the bigger particles.
Thank you. Another question, do the particles have the potential to create other ROS in indoor air?
Other ROS, yes, I think if they are basically, the way I try to explain it, the particle has to be redox active, meaning it should be able to transfer the electrons from a donor to oxygen. I think that if it has that capability, then yes, it will generate ROS. So like compounds such as quinones, metals, they are very efficient, in doing those. So yes, they can generate definitely other ROS, such as organic peroxides also, if they have this capability. So absolutely, yeah.
Another question, are DTT measurements enough to assess the oxidative potential, or would performing separate measurements of ROS give more insights?
Yeah, by no means I imply to say that DTT measurement is good enough, and that's why we use a battery of these assays. In our recent publication published in ES&T, we showed that some of these assays are very relevant, some of them are not so relevant. I think we found out DTT consumption and hydroxyl radical generation are important. Maybe glutathione consumption is not so important. Why? Because the way we measure the glutathione consumption in a chemical assay does not represent the cellular condition. But the cellular ROS measurement is the most relevant, because it incorporate other mechanisms as well. So yeah.
And one final question, there are a lot of discussions on the role of interface in multi-phase chemistry. Assuming the same composition is there a particle size effect on DTT responses?
Yeah, so as I said that, the way we are measuring the DTT and the ROS consumption, what we do is we collect these particles, we incubate them, like we extract them into water. So we are removing the effect of the size because everything will, becomes suspended in the water, um, and then solubilize most of it. So the size which is present in the ambient air, that information is lost because we now have a suspension of the particles. In the real exposure, I think yes, size plays a very important role because I think that's where maybe we should use more devices such as air liquid interface. We are not using those. So in that way, size plays a role, but not in our construct.
Okay, great. So I'd like to conclude our webinar today and thank Professor Varma again for his excellent presentation, very informative. A lot of great work both on outdoor and indoor air pollution. So very relevant to our research here at Purdue University. And I think, Professor Varma, you're really taking a big leap forward here in trying to understand how particles in air affect our health. So thank you very much and really enjoyed your talk.
Thank you. Thank you so much, Brandon, again for inviting me and hosting me for this wonderful series of the talk. Yeah. Thank you again.
Thank you.
Bye, everyone.